next generation sequencing. The platform prices and entry points have come down quite a bit, and there's a lot of new technologies. And so it's become very routine and available, especially in the area of diagnostic investigation. We've all seen this kind of with COVID, is that sequencing is becoming more and more sort of the currency of these investigations, and the methods are becoming more rapid. And so we've been using primarily Illumina-based technologies as well as nanopore, and we kind of integrate our next-gen sequencing originally mostly for pathogen discovery and characterization. But the tools that we've developed are pretty standard across the board for the most part. So we have an alignment-based analysis pipeline, and this is pretty similar to what a lot of other labs sort of do. And there's various flavors of this in the bioinformatics world, where basically you're identifying viral agents using your raw sequence data and then mapping it to a known genome. And so our pipeline, it's written in Python, and it's got SQL based data management. So pretty standard methodology. And so just for a quick overview, you have raw data, your sequencing data comes into the pipeline, it's quality filtered and then converted into a blast capable database. And then it's cross-referenced against these viral reference genomes. And so then what you get is this nice visual output of the reference genome compared to your reads that map it. And so it's a pretty straightforward tool. And the nice thing is with the SQL interface, you're able to store all your subsequent data for any downstream type of analysis. And like I said, it's a pretty standardized tool. It works in most cases. And it's usually sufficient for the identification of the genomic sequence of whatever pathogen you might be looking for, and can be used for any subsequent characterization. Rarely, though, we do have these situations where we get cases that come in that are PCR negative by our diagnostic testing, but virus isolation positive. And so in these situations, we're not getting good assemblies to the known reference genomes. And so these tend to turn into situations where we're trying to perform more pathogen discovery. And in these situations, reference-based methods tend to not work as well. And so an analysis method that doesn't require the direct sequence of alignment to a reference genome could be more effective for novel pathogen discovery. And so this is kind of where machine learning comes in. And the particular area of machine learning, because it's actually a very broad field that we were interested in leveraging, was neural networks. And these were conceptualized back in 1943, originally by Warren McColgan and Walter Pitts. So these have been around for quite a while, but it's only been recently that we've really been able to leverage these technologies, given the expansion of computational power through devices such as GPUs, which are able to perform extensive parallel math. And then given that we have these massive data sets available now, generated by tools like our sequencers and various devices, it's really kind of sort of becoming, we're getting into the golden age of machine learning as far as being able to really leverage it now. And so what machine learning is, in particular neural networks, these are universal function generators. So where the traditional approach to programming is sort of you define the steps to an outcome. So you write a series of conditionals and questions that sort of walk you to the output. Machine learning kind of flips that approach around. And instead, what you do is you define the outcome, and then the model learns the steps to get there. And for the most part, it pretty much outperforms most other models, given that you have decent data and enough of it. And so what we kind of conceptualized with this was a system where we could take a sample, perform next generation sequencing on it, and then actually, instead of looking at the sequence per se and aligning it to something, convert the raw data into an image, almost a pattern, so to speak, and then teach a machine learning model to recognize those patterns of particular viruses at different taxonomies, and then be able to say from that pattern that it's a particular viral member or related to something. And so in order to get to this, what we did was we developed 4 individual software tools that perform the following tasks. First, they produced image data, we took image data from the raw sequence and then built the convolutional neural network model. And so the first tool was able to convert the viral FASTA sequence. So this is the raw, or this is the viruses that are known into that sort of image domain. And then the second tool would actually build a convolutional neural network around that data. And then the third tool converts the raw sequence data that you generate on your sequencer back into that image domain. And then the fourth tool would recall the neural network and then make the predictions between the two. So you have the patterns that are known of different genera, families, and species. And then you have the raw data coming off the sequencers. And so to sort of give a better idea of this, in order to do this, we have to be able to convert our sequence data into an image. So how do we do that? Well, it turns out we had developed a tool previously that was sort of perfect for this purpose. We actually took the features from pan-viral microarray, and these were based off of 60 nucleotide features that were determined to be conserved between at the family level, the genus level, as well as the some species, although it was primarily targeted at family and genus. And so what this ends up giving is similar to a microarray. We have 10,816 unique features, which gave us 104 by 104 pixel grid. And so what we do is we then cross-reference our sequences up against this grid. And so what you end up getting is sort of almost a virtual microarray out of the sequence comparisons. And so the neural network is able to learn these patterns. And this isn't just a direct nucleotide alignment. We actually do a 6-frame, it's called T-BLAST-X. Basically, it's a translated nucleotide sequence alignment to the pattern. So it allows the network to be able to sort of generalize on these and pick up more potential features than it would were we to do just a direct nucleotide alignment. And the sort of the color intensity you see is based on the percent identity of the translated alignment. So that's where we sort of generate our sort of image from raw sequence data. And as you can see, here's 6 different images from different viruses, and the patterns are clearly unique. So the first one's classical swine fever, second one is Seneca Valley virus, third one's a West Ebola virus, fourth would be variola virus, human enterovirus, and duck hepatitis. And these are all sort of virtual, so we didn't work with these viruses per se. These were generated off of data from GenBank. And these were sort of our sort of reference patterns. And so in order to then compare whether or not the model worked, we ended up using foot and mouth disease virus, because what we wanted to see was how sensitive are we to be able to detect the presence of a virus using this compared to the direct alignment method. And so in this figure, what you can see is up to 20%. What we did was, and just for reference here, we did 20 replicates, and what we did was over increasing amount of genome variation. So we were randomly introducing variations into the nucleotide sequence up to 0 to 50% of the genome. And so, and these were completely random. And so up to 20% variation, what you see is both the direct alignment method and the neural network model are both able to identify the agent as foot and mouth disease virus. Now, once you hit 30% of random variation of the genome, the direct alignment method drops to about 55% able to identify the agent as FMD, whereas the neural network at 30% variation is able to 95% of the time identify that the agent present is FMD. At 40%, you've dropped to 5%, basically only 1 out of the 20, it's still recognized as FMD, whereas the neural network model was able to do it at 10%. However, one interesting thing about the neural network and one of the advantages is, even though it didn't detect FMD, it did believe that it saw a virus similar, a picorna type virus. And so this is sort of where the sensitivity comes in, is that it's able to detect other picorna viruses potentially, or that there is the presence of a picorna-like virus in the sample. And then at 50%, both models basically fail. So really the significance comes with the neural network model as far as its increased sensitivity, right around the 30% mark. So based on this, it's clear that we do have an improvement in the ability to detect the presence. So the neural network approach does outperform the direct alignment approach, at least at determining the presence of a virus. However, the alignment approach is still going to be better at specific species identification, given the resolution of the pattern and the way that the model is trained. And in diagnostics, the neural network approach is really complementary to the alignment-based approach and increases the sensitivity of NGS for novel agents. So we don't really see this as a system to replace alignment-based methods, but in the event that you need something that can potentially detect something, it definitely is something that you may want to run in parallel with. And so our goal is to produce a artificial or a machine learning system that can rapidly identify the presence of novel viral agents with multiple viral genera. And so ultimately what we want is not just a single neural network model, but multiple neural network models that work in parallel, each one sort of specialized for its either family or genera of viruses. So once you create that one image of off of your raw sequence data, you're able to then have each individual neural network look at that image and say, I detect a picorna virus in this part of the image, and then, whoops, sorry, moved too quick there. And then it moves on to the next one, and it detects, and it says, oh, well, I see a porcine endogenous retrovirus in the pattern, doesn't see anything in Filoviridae, sees CSF in that part of the pattern. Holy cow, these pigs are really infected. And then no paramyxovirus, thank goodness. So if you can sort of see the idea of this, is it's very rapid, because once that image is generated, you have all these specialized neural networks that can kind of look at it and determine if there's potentially an agent present. And so that was kind of the sort of one part of our area of development. We've got a few different projects looking at machine learning in the lab. One other area that we've been looking up is working at integrating machine learning is kind of more the proteomic side. And so with this, what we're looking at is we're using MALDI-TOF, looking at the spectral data for applications of machine learning. And so the part of the reason that we have an interest in this is while we do tend to do primarily next generation sequencing, and it is sort of our routine method for characterization, it is time consuming, and it's actually, as many know, it's pretty expensive with regards to consumables and cost just to run the sequencers and maintain them, even with the sort of reduced entry point. And so for those of you who aren't familiar, MALDI-TOF is also known as matrix-assisted laser desorption ionization time of flight. It's a form of mass spectrometry that can be used to generate unique spectra from samples containing pathogens. It's cheaper than next generation sequencing. Basically, you're doing your initial sample prep, you're ionizing sample, and then you're generating a spectra. And it's quite a bit faster than the current NGS methods. So even though NGS is becoming much more rapid with a lot more capability in real time, it at the moment, it still is more time consuming than the MALDI-TOF sort of prep. Currently these methods are more common in bacteriology, but they have been proven in some publications to be effective for characterizing viral agents as well. And so sort of for this project, what we are conceptualized and are working on is generating primarily initially for proof of principle. We have VI cell culture that we of our pathogens of interest. We perform the Multitoph spectrometry on it. We're able to compare the cell lines and generate spectra for the agents that we can then generate basically a database of these spectral references. And then once we have these, train a machine learning model again to be able to recognize these sort of patterns and then be able to identify a specific agent from within those spectral references. And so, again, this was just a quick overview of this area of the project. The spectral libraries representing the fat agents could be used to perform the sort of valuable diagnostic screening rapidly. Again, it's more cost effective than next generation sequencing, and definitely more efficient and for preparation and analysis and faster results. And it's also useful for guiding follow-up testing analysis and determine whether or not a particular sample may want to be looked at for next generation sequencing. And so I know that was a lot in a short amount of time, but I'd like to thank everyone that obviously at FATL that have supported me and I've worked with over the period of time. I will move on and we can take any questions.
Thank you, Dr. Barrette. That's an excellent presentation. I think gives an overview on perhaps some of the really exciting tools, analytical tools that are now being put together with next generation sequencing. Perhaps while people are thinking about questions, can I perhaps ask a sort of a generic question as a starting point, which is thinking about these virus discovery pipelines. How analytically sensitive do you think they are? And I'm thinking in comparison to other methods such as a targeted PCR or an immunoassay, for example.
So the alignment based, and this is actually a question that we've been interested in resolving in the lab, because you know, a lot of it's going to depend on one, the platform, to some degree the chemistry you're using. You know, you've got systems like the Prometheon, which can generate a massive amount of data. It's like 12 to 15 terabytes over I think like a 48-hour period. So in a situation like that, granted you're trying to find potentially a needle in a haystack, it's sort of a moving target. I think that the sensitivity of next generation sequencing is probably at some point potentially could surpass PCR, but a lot of it is going to depend on one, how good the sample is, how where the target is on the genome, and you know, it's we're not there yet, obviously. I mean, I would say that there's a lot of potential, but at what point is it diminishing returns? I mean, how long do you want to run a sequencer to be able to detect something that may be more difficult? And we've had situations like this where degraded samples did not perform well in PCR, but when you do small fragment sequencing, you're able to sort of piece together enough of the genome to be able to detect it. It doesn't happen often, but we've had some cases where we've been able to do that with degraded samples.
Thank you for that. So I suppose thinking about your talk and the pipeline that you've described in your talk, can you perhaps comment on how well it's able to detect individual isolates in a mixture? So if you've got a mixtures of sequences together, can it distinguish between those different species?
So the way that it's currently designed, each individual neural network that's sort of specialized into either a family or a genus will find a individual highest probability agent within its taxonomic level. So if you have 2 picornaviruses, it's going to detect the predominant isolate as the highest probability, but it will probably have, it also will report some probability for another potential picornavirus that might be similar. And so when you're dealing, if you've got a situation where you have 2 picornaviruses, it's going to struggle to identify that. And at that, you almost would have to follow up with an alignment type method where you sort of align to the predominant strain and then remove the data from that and then try to perform a subsequent assembly. So it will give you an indication that there's probably something there, but you're going to have to follow up. Now, with regards to different genera, it should be very easy, because if it makes a call on say, classical swine fever and one on foot and mouth disease, those 2, because they're separate genera, they're very easy to distinguish the neural network. You have pretty good confidence that both are co-infection. So it does have some difficulty when they're within the same taxonomic level, but it should give indications that that's the case.
Yeah, thank you for that. There was a number of related topic and related questions raised in the Q&A box, and thanks to everybody who's raising the questions. And please don't feel shy. Keep typing in some questions for our speakers. So thinking sort of about another technical challenge, and perhaps we've certainly had some experience of this here locally using these virus discovery pipelines. How do these systems work when there's actually no virus present in the sample? Does it falsely identify the presence of a virus that's actually not present in the sample?
And so that is one of the areas that we sort of learned the development of this that was problematic, is that you actually have to train the model to recognize a no virus situation, because if you don't do that, it will, the default is when you train these neural networks on all these different viruses, basically you're telling the neural network there's always a virus, tell me which one it is. And so you actually have to go through this process where you do end up having to train the model to recognize a no virus situation. And that's actually a lot more complicated than training for a virus, because with a virus, you have some sort of generalized patterns. They tend to be related with some variation. But with no virus, you tend to have a very sparse array that the virus, that the neural network is trying to learn to recognize that it's sort of the reverse of what it's really being trained to do. And so it's actually a very complex task, and we do go through that series of training with each individual model, where it has to learn sort of a no virus is a condition. And so that is a situation that comes up as a probability, regardless of which model it's trained on or is being trained.
Yeah, thanks. We had a little bit of experience of that, as I say here, where we had collected some samples from a wildlife species in Kazakhstan, which of course is a landlocked country in the middle of Asia. And when we put it through an NGS pipeline, we actually got a load of aquatic marine viruses, which seemed to be not very consistent. And so we had to work through some of the problems that you're describing here, which I think is one of the learning experiences, I think, from using these types of pipelines is understanding the limitations and the extent to which they can falsely identify pathogens. So we have another question from the floor, which is, it says, in my experience, T-BLAST-X is very computationally intensive. How difficult would it be to combine a program like Authfinder before the multiple alignment step to use a more rapid and efficient program like T-BLAST or BLAST-X?
And that is where the primary bottleneck is. It would probably be, as long as it's a command line based tool and sort of works through a local database with to perform that sort of comparison, it should be fine. Actually, in our discussions, that's been our biggest bottleneck for the timeline. So when the process happening is happening, the slowest step of the entire process is that image generation. And it's because T-BLAST-X is such an inefficient algorithm. So really being able to modify or put in a more efficient algorithm to that would improve our situation greatly. So right now, to create an image off of 50,000 sequences on my laptop, it takes like 60 seconds, which that's the longest step of the whole process. Once that image is generated, the neural network's like that. It just looks at it and says, oh, it's this. So yeah, that is where our bottleneck occurs. So any tool that could sort of supplant T-BLAST-X and be more efficient would improve our fortunes in that regard.
Thank you. I think we'll have to bring your Q&A to an end. Thanks to everybody who's posted questions. I'm hoping that we'll have time to pick up on a lot of these questions in the, as I say, in the roundtable discussion after the 4 speakers. So thanks, Dr. Barrette, for an excellent presentation. Thank you. So for our second talk today, it's from Dr. Kylene Kehn-Hall. And so Dr. Kehn-Hall is a professor within the Department of Biomedical Sciences and Pathobiology at the Virginia Maryland College of Veterinary Medicine, as well as the Center for Emerging Zoonotic and Arthroborne Pathogens called CZAP, which is a great name. Research in our lab is centered on Rift Valley fever virus and alpha viruses such as Venezuelan, Eastern and Western equine encephalitis virus. And more recently, she's been involved in projects looking at SARS-CoV-2. So today she's agreed to provide a talk on the use of nanotrap particles to enhance viral diagnostics and detection. So Great. The floor is yours. Thank you.
Thank you, Dr. King. And I also want to thank the organizers for giving me this chance to talk today. As Dr. King mentioned, I'm going to be talking with you all today about some work that my lab has been doing for approximately 10 years now using nanotrap particles to help with viral diagnostics. And so all of you know that kind of the bread and butter of diagnostics, especially in terms of viruses, falls within these kind of 4 techniques listed at the top of the slide here, where we can use ELISAs to look at specific viral antigens or look at antibody responses, or we can use molecular methods, including PCR-based tests, or even some of the next generation sequencing techniques that we'll hear about today. Or, if we want to actually further characterize the virus, we can isolate it and try to grow it up in culture, but that of course takes quite a bit of time. In terms of challenges, and really all of these methods have a number of challenges associated with them. We know that transport and storage of samples can greatly affect diagnostics. So if the sample is not kept or not, the cold chain is not kept, then you can have degradation of your analytes. We also know that cases are often recognized late after infection, and in case of many viral infections, especially acute infections, they may have decreased viral levels or detection of the virus is no longer possible. And all of these assays have their own sensitivity issues. We're also very interested in trying to diagnose somebody pre-symptomatically. So can you tell what pathogen they have before they even come down with symptoms? And so we asked the question early on if nanotrap particles could help us with these challenges. And so I'll start off with giving you an overview of what the nanotrap particles are. This work has been done in collaboration with Sirius Nanosciences, which is a spin-off company of my former institution, George Mason University. And these particles are essentially core shell hydrogel particles. And so the nanotrap, kind of the core part of the particle, is made up of the hydrogel. So it's a polymer chemistry, and the material is very similar to what you find in contact lenses. And the affinity baits are coupled with this nano core. These are chemical dye baits that are typically used in pathology analysis or even the textile industry. So these are things such as Seabricon Blue or Reactive Red 120. And there's an optional size sieving shell that can be added to the nanoparticle. And the purpose of this shell is to actually exclude high molecular weight analytes. So these particles were initially developed for protein biomarker discovery. And so the idea was that the analytes would be taken up inside of the nanoparticle, bind to the chemical diabates, and high molecular weight proteins such as albumin would be excluded, thereby increasing the sensitivity of the assay. But over the years, the particles have really been developed further, and they're quite customizable. And so there's three kind of main varieties that they come in right now. There's the Nanotrap core particles, which are typically the ones that we use. These are used without the size-saving shell, and these are in applications where what you want to capture is larger than the nanoparticle of interest. So these nanoparticles are around, maybe 400 nanometers in size. And if you're looking at capturing viruses, bacteria, or even exosomes, you want to use this particle. As I mentioned, the core shell particles are going to be those that you want to use for smaller analytes, where those analytes can actually be captured within the particle. And luckily, in more recent years, magnetized versions of these particles have become available, which really enables quicker processing. So previously, the particles were taken out of solution by centrifugation methods. And so we've been working with these magnetic particles for the past couple of years, which really makes our life a bit easier. So the technology is based on, I would say, three main principles: the ability to capture, the ability to concentrate, and the ability to preserve. And this preservation aspect really makes these particles unique, especially in a diagnostic or a detection capabilities. Analytes, you really can capture most any analyte you can think of. As I mentioned, the particles were originally developed to capture proteins, but my group has worked on optimizing these particles for virus capture. You can also capture metabolites, nucleic acids. There's been some work done with exosomes, and the sample types can be quite varied. So even blood. So we've done a lot of work looking at blood, not serum, but actual blood, which is a challenge to work with, but we've been able to work out protocols. Urine, nasal fluid, saliva. You can do multiple sample types, and the applications are really whatever you can imagine. So you can use these for discovery applications, diagnostics, or surveillance. The workflow is quite simple. So you take your Nanotrap particles, you add them to your sample of interest, and the volume of your sample can be varied. You can, if you only have a small amount of sample available, that's fine. If you have a large amount of sample, or you need to dilute your sample, that's also fine, because essentially the particles are going to soak up your analyte of interest and help to concentrate them out of solution. And so what's shown here in this graphic is the Nanotrap particles are able to take the B, which stands for the biomarker here, out of solution, and they will exclude things like the albumin from the nanoparticles. And then you can pull them out either through centrifugation or using a magnet, or there's even filter-based methods. This allows concentration of your analyte, and you elute them off of your particles, and then you put them into your downstream assay. And so I want to emphasize here that this is really a sample preparation technology. So the nanoparticles themselves are not going to tell you what analyte you have in your sample, but rather you need to couple it with standard diagnostic methods or detection methods to tell you I have virus X in the sample. And you can really couple it with many different types of assays. This has been done with immunoassays, PCR-based assays, mass spec, lateral flow assays, etc. So our first project wanted to see if, or aimed to see if the Nanotrap particles could be applied to enhancing Rift Valley fever virus detection. And when we first started this project, we used some spike samples where we added virus to either bovine serum or sheep serum, and plus minus nanotraps, and we looked at the amount of virus present by RT-qPCR, and you can see that in the presence of nanoparticles, we see about a 10-fold enrichment of our genetic material detected. We were also interested to see if the analyte we were capturing was the virus itself, and would that virus maintain infectivity. And what we did here is that we added our Nanotrap particles to our virus, and then we did a plaque assay. And what you can see is that there is actually infectious virus being captured by these particles, and we can basically loot it off by adding it to cells. And in the case of our nanotrap particles, we have about 10-fold enrichment of the amount of virus detected. Interestingly, we also saw that incubation with the nanotrap particles preserves viral infectivity, so that if you incubate samples at either room temperature or 37 degrees at various time points, always, as you can see here in gray, the gray bars, we see increased viral infectivity with the nanotrap particles, even at 37 degrees Celsius, indicating that the particles can preserve the viral infectivity. And this is pretty interesting when you think about it from a sample transport point of view, that you could add particles and potentially transport your sample without cold chain transport. These particles were initially designed to capture protein biomarkers, as I mentioned, and they have great protective ability for protein biomarkers. So we aim to identify if these particles will also be protective of nucleic acid, specifically RNA. And so in this experiment, we spiked purified viral RNA into a sample with or without RNase A. And you can see that in the presence of the nanotraps, the viral RNA compared to the control is protected from degradation. And so this is another aspect of the particles really preserving that viral infectivity. And we think that the mechanism which this is occurring is either the RNase A is being excluded from the particles, which is one possibility, although it's not likely because its molecular weight is quite small. Or perhaps if it's getting taken inside the particles, it's spatially distant from the nucleic acid, or it's rendered enzymatically active by binding to the chemical weights. We also were interested in looking at protein detection with these particles. And for this, we decided to look at the nucleoprotein of Rift Valley fever virus, which is the most abundant antigen and also can be detected outside of the virion. And we did a quick screening of different nanotrap particles. Our detection here is just a simple Western blot. Compared to our input, you can see that our nanotrap particle N245 could bind to our nucleoprotein the most efficiently. And if we look at samples that contain 20 microgram per mil of nucleoprotein, which is below the limit of detection, if we do this in the presence of nanoparticles, in all cases, we can detect nucleoprotein in different serum samples. We've also looked at this in terms of respiratory pathogens, and specifically, we were very interested in looking at influenza viruses. We did a screening of nanoparticles and saw that the genomic copies could be detected, and with NT46, we see about 10-fold enrichment of influenza A H1N1 virus. We see similar enrichment of another influenza virus H3N2, as well as influenza B virus. If we look at this in a more clinically relevant matrix, a lot of the previous experiments were done in cell culture media. We can still detect enrichment with the nanotrap particles. So we did 10% saliva from 3 different donors, and you can see we have at least a 10-fold enrichment of our viral genetic material. The nice thing about the nanotrap particles is that you can dilute your samples. So if you have a really complex mixture, you can dilute them to enable the particles to bind better, and it still is able to work with nanotrap particles due to their concentration ability. We also perform testing in 100% saliva, and we're still able to see a very nice enrichment with the nanotrap particles. We've always wondered about how the nanotrap particles were binding to our viruses, and we hypothesized that it was due to interaction with the surface proteins of the virus. And for influenza virus, the most prominent surface protein is the hemagglutinin HA protein. And so to start to address this hypothesis, we incubated purified HA protein with our nanoparticles, again, did a screening, and this is our HA detection limits without nanotrap particles, and then NT46 captured HA most efficiently. So confirming that it can bind to the surface proteins. We did a dosing study, and we can see that at all doses of HA, when it's not detected without the nanotrap particles, we can detect it in the presence of nanotrap particles, showing we can get 100-fold enrichment with these particles. We also extended this to look at multiple influenza HA proteins, and in all cases, we could see an enrichment with the nanotrap particles, whether it was H5 viruses or H7 viruses. And so this is pretty important because it suggests that if the virus mutates, we can still bind to that virus and enable detection. We've done quite a bit of work looking at the impact or the use of nanotrap particles for arboviruses. And so in this project, we were looking specifically at its ability to capture dengue, Zika, and chick. And what's interesting about these particles, or excuse me, these viruses, as well as many other pathogens, is that they don't always occur in isolation, and you can get co-infection scenarios. And so in this project, we looked to see if we had two viruses present in a sample, could we still detect the virus? Would the particles still be enriching in the capability and the capacity of these co-infections, or would there be some sort of competition between them? And so here we spiked in dengue and Zika, and you can see in both cases, we're still able to enrich with our nanotrap particles. The same with chikungunya and Zika, we still see enrichment, as well as with chick and dengue. And so this indicates that the particles are not saturated and they're still able to bind to multiple pathogens. And so over the years, as I mentioned, we've done this for a number of different viruses, which are shown on the left-hand side of the slide, many different arboviruses, as well as respiratory pathogens. And we still didn't have a great idea of how the particles were binding to the viruses. But we were, in recent years, we were able to do some electron microscopy where we were able to see the viruses binding to the surface of the nanotrap particles. And so in this TEM image, you can see the nanotrap particles are in black, and the virus, in this case, Venezuelan equine encephalitis virus, is in gray, and you can see clearly it's binding to the surface of the particle, and it actually tends to aggregate around the particles. Now, all of this work really set us up when the pandemic hit to be able to jump in and help with the SARS-CoV-2 diagnostics. And so we applied all that we knew, especially what we had learned from the respiratory pathogens, to be able to enhance SARS-CoV-2 diagnostic methods. And then so in this project, which was published late last year, not only did we show we could enhance viral detection, but we also developed a quick workflow which could basically eliminate RNA extraction from the workflow. So here you just have your sample added with nanotrap particles, you concentrate them out of solution, and then you lyse them quite rapidly using a heat and a detergent extraction method. Remove your particles and go directly into PCR. And when we compare this to a traditional extraction using QIAGEN kits, which we typically use for this process, you can see that with the nanoparticles, we always get a decrease in CT values compared to without. If we do this with virus spiked into transport media, or if we look at virus spiked into saliva samples. We've also were able to show that the nanotrap particles could increase the sensitivity or the detection in diagnostic remnant samples. So we're able to get these where there are some samples that were previously tested positive, whereas there's others that tested negative. When there's high level of virus present, with and without nanotrap particles, we get similar CT levels. But at low viral levels, which would be as expected, we get an enhancement of virus detection. And even samples that previously tested negative, we were able to detect virus in the presence of the nanotrap particles, both here and in this set of samples. And then lastly, we're really excited about the potential of using the particles for pooled samples. And as we know, with the COVID pandemic, there was just an insane increase in the need for diagnostic assays and that the community needed to try to keep up with. And one potential solution would be using pooled samples. And so in this diagram, it's just an example where you could potentially pool 10 nasal swab solutions together with one of them being positive, and you can test them in concert. The concern here is that if your limited detection is not low enough, that you could miss out on some positive samples. And so to sort of address this, at least in kind of a mock scenario, we took a positive sample, diagnostic sample here, diluted it at 1 to 50 or diluted it 1 to 100, and added and analyzed it with and without nanotrap particles. And with the nanotrap particles, we could increase our diagnostic sensitivity. So this really opens up the floor for us to use this method for pooled testing in multiple different diagnostic avenues. So with that, I'll just stop and acknowledge my collaborators. All of this couldn't be done without my collaborators at Sirius Nanoscience. I primarily work with Ben Lupine, but there are many members of Sirius that are really fabulous to work with. In terms of my lab, Dr. Shafagati really led this work during her PhD. And then in more recent years, Chow in the lab and Ivan in the lab has spearheaded these efforts. And we've received funding over the years from many different organizations, from Institute of Infectious Animal Diseases, DARPA, DITRA, and then more recently from the Schmidt's Future for our COVID work. That, take questions.
Thank you very much, Dr. Kehn-Hall. Very, very clear presentation, very exciting new applications there, I think, that perhaps we can all relate to. So while people are typing in questions, perhaps you could just tell us a little bit more about any specific applications that you see the use for these nanotrap particles might be for the veterinary sector and veterinary diagnostic sector.
Sure. Yeah, great question. So I think the last slide that I ended on really speaks to that in terms of pool testing. I mean, so let me back up and say that any current, I would say, veterinary diagnostic testing, I can see that the nanoparticles could really be coupled to. And so because it's so versatile that you could couple it with any sort of molecular based testing, or even we've done work looking at lateral flow assays. But I think the pooled method has a lot of potential, because I can imagine if you're screening herds, for example, and there's large number of animals that need to be tested, and you want to keep costs down, if you can pool samples together and then put them through a molecular test or an antigen test, you could really speed up the processing as well as keep costs down.
And can I just, from my own information, can I just ask you two questions? The technology itself is owned by a commercial company, is that right? And so are there any IP issues relating to the use of this technology for different diagnostic applications? And do you have to work closely with the company, or is it an open platform that you can use for lots of different applications?
Sure. So there's two different ways you can go around it. They actually commercially sell the particles now. Initially, they only sold one type, but over the years, working closely with them, we've developed them as a kind of a viral diagnostic or viral capture kits. So you can buy the viral capture kits directly with them or from them, or more importantly, I think probably the better way is to work directly with them. And so they can work very closely with you and help you optimize the particles for your diagnostic assay. There are, I'm sure, IP issues involved there, but they've worked closely with many different academic units as well as labs. In fact, LabCorp right now has EUA authorization to use the nanotrap particles for COVID testing. So they can work very closely with all sorts of different organizations.
And the costs always come back to the costs. What are the costs of using nanotrap particles as a way of enriching samples, say in comparison to nucleic acid extraction or a monoclonal based immunocapture based method? How do the costs stack up?
Sure. Yeah, I don't have a great exact cost for you, but I would think that they in general would be less expensive than a nucleic acid extraction kit. So if you can just do that simple heat and detergent based solution method, they would be cheaper than your standard nucleic acid extraction kit.
Okay. And there's a couple of questions here from the chat box. So one asks, for the use of the nanotrap particles in the field, followed by sample transport, can the particles withstand freezing or extended 4-degree storage? If so, for how long?
Mm-hmm. I don't believe they can be frozen, but 4-degree storage is fine. So they can be stored at room temperature, or they can be stored at 4 degrees, and they can be stored, I want to say, months. I know that the company has done extensive testing on the shelf life of their products, but I'm not really involved in that aspect of that. Okay,
and there's a second question from the floor as well, which perhaps links to what I was asking before about the applications for the veterinary sector. And so you mentioned the use on saliva. Have you done any work with oral fluids that could be submitted for PCR testing?
Yes, the only work that I've done with saliva is looking at detection for influenza and then for coronavirus. So those were for the most part, spike samples, but we've also, as I mentioned, had some diagnostic remnant samples that were given to us, saliva-based. So it definitely can work with saliva, and I think that could be an important application.
And there's sort of a follow-up question, which is, how well does this method work with complex sample types, such as fecal material, where there would be perhaps a lot of host material, as well as other bacterial contaminations that wouldn't, bacterial contaminants, which wouldn't be necessarily what you'd want to be picking up in the assays?
Sure, great question. So the particles are pretty good at helping to simplify a sample matrix. There's some kind of tips you can do where you can, as I mentioned, you kind of dilute out the sample a little bit. We had to deal with this quite a bit when we were dealing with whole blood samples in order to try to still get binding of our virus in the presence of whole blood. But I imagine, again, bacteria would be a major problem in the fecal samples. We have done some work with my collaborator, Dr. Monique Van Hoek at George Mason, where she's looked at the ability of bacteria to bind to the particles. And you can get some binding, but they don't bind that great to the particles. And so in that case, that might be a good thing if you're looking at a viral pathogen and you're trying to simplify basically your complex, your fecal mixture. You could hopefully wash away some of that bacteria from the nanoparticles in order to simplify the sample and get it into your downstream assay.
And in terms of using the materials that are enriched on the nanoparticles, how do they work for, if you link to the previous talk, we heard all about virus discovery and next generation sequencing. Are they suitable for sample prep for that?
Yes, I would say the short answer is yes, although those studies are ongoing. So I haven't personally done any coupling with NGS, but Series Nanoscience has some promising results, especially if you're binding to entire virions, which is what we see. The material is quite protected, and then it can be used for sequencing applications for sure. And I know there's some other individuals who are exploring that in the academic sector.
Thank you. I think probably we need to draw your question and answer to an end, but I thank again everybody who's posting the questions in the Q&A box. They're still coming in, and like I said earlier, we'll try to pick up on them in the roundtable session later on. So for the third talk today, I'm pleased to introduce Dr. Christian Happi, who is the director of the World Bank funded African Center for Excellence for Genomics of Infectious Disease. And he's professor of molecular biology and genomics and the former dean of the College of Postgraduate Studies at Redeemers University in Nigeria. And so Dr. Happi has extensive experience of infectious diseases, particularly genomics, and including a range of diseases such as Ebola and Lassa Fever. And so today he's going to provide a presentation, and I think perhaps quite an important topic, of genomic characterization and surveillance of microbial threats in West Africa.
Well, thank you. Thank you, Don, for the opportunity, and then thank for the organizer to give us a platform to make this presentation. So I will really go on and then just talk about what John just introduced right now. So I just want to get my little pointer. So I will, my presentation is actually genomic characterization. The Africa Center of Excellence for Genomic of Infectious Disease is a World Bank regional reference laboratory in Africa for infectious disease genomics, and is also an Africa CDC under the Africa Union reference laboratory for infectious disease genomics on the continent. And as why do we use genomic as part of a way to kind of change the narrative as a way we study infectious disease in Africa, and especially do surveillance? So simply because, genomic sequencing, has revolutionized the way we approach healthcare and research, but many of the technological and health benefits do not really reach Africa because simply we do not have the platforms and then also the know-how in order to do that. And as such, we created a platform called the Africa Center of Excellence, which is what I call a consortium of academic and clinical testing centers in West Africa with institutions like Harvard and then the Broad Institute and Dumina in order to actually transform and then do transformational impact for genomic surveillance on the continent. And then the overarching goal is actually to create a vibrant academic and research environment that transcends national boundaries in order to conduct what I call responsive, highly responsive, ethical, and high quality translational genomic research in Africa. So you'll see that in the next few slides. But then ultimately, we think that we can use this platform to build the necessary capacity that Africa needs in order to use genomic-based tools for prevention and control of infectious disease generally. And in order to do that, we basically just went on not only to look at the fever of unknown origin on the African continent, but also went further to start applying these new technologies for zoonotic diseases and then start looking into animal health. So, and ultimately the goal is basically to create the next generation of African pathogen hunters in the dead horse of Africa, that can start using genomic knowledge in order to hunt for this pathogen and then characterize them before they start hunting us, simply because the global community and in the world has always been playing defense, when it comes to pathogens. But I think with the knowledge and skill that we have now, I think it is high time for us to start playing offense. And as such, we think that through this platform, we first had the first demonstration of genomic, transformational outbreak response in 2014, during the Ebola outbreak in West Africa. So when this outbreak happened, we were able to use this information in order to respond to Africa genomic needs. And then as such, when this happened, when Ebola happened in West Africa in 2014, 2016, so we were able to first use that demonstration, and show that in action. In the first week of the outbreak in 2014, we identified 35 cases, confirmed, 14, but absolutely used genomic data as a way to trace, the first index case to a traditional healer in West Africa, to which, to Sierra Leone, where to which all these cases were spreading. And more importantly is the fact that, when this happened, we actually pioneered what we call open data access and open data sharing. We are very happy that pioneering this in 2014, we generated the first 99 genome, full-length genome of Ebola during the first 3 weeks of the outbreak in West Africa, made this data available to the international community so that those interested in developing diagnostic, vaccines and therapeutics could use this information. But more importantly, actually developing this method for next generation sequencing, we shared it, to the international community. So we're very happy today that the global community has embraced what we started in 2014, to GSET, where everybody is depositing, SARS-CoV databases. But what was unprecedented in what we did was actually from sample collection to when we deposited the data in GenBank, we actually did this in 10 days. And really our paper could come out only 4 months after when we did this exercise. So the most important thing for us was actually to save lives. So, but then when we realized that, diagnostics, through PCR and then also through next generation sequencing really was not the way to go, we translated, the genome sequence data into the first, to rapid diagnostic tests. And then through collaborators, we were able to generate the first Ebola rapid RDT that was approved by the WHO and then the USDA for, under emergency use authorization. So, and then the introduction of this test in West Africa at the time really was a major turning point in order to contain this outbreak. So moving forward, prior to Ebola outbreak, we're working on a disease called Lassa Fever. And this disease has been discovered in Nigeria about 52 years ago, and up to 2008, there was no diagnosis on the ground. So we performed the first the first extensive genome sequencing of this virus, characterized it, and then we were able to show clearly that this virus, even though it was described 50 years ago or 52 years ago, emerged from Nigeria to present day Nigeria about 1,060 years, was circulating within Nigeria for 600 years and moved westward, about 400 years ago. But then using again the same approach that we did for Ebola, we were able to use that to generate what we call a pan-Lassa rapid diagnostic test. The reason why we talk about pan-Lassa rapid diagnostic is because the Lassa fever has several lineages. So we're able to generate, a test that can actually capture all of the different lineages circulating. And that really, again, is this antigen capture, a lateral flow rapid diagnostic test will also generate an ELISA. So, and then the sequence of this virus, that the first catalog of this virus that we sequenced, accelerated, development of vaccine for this disease that was ignored for many years. And then we're very glad today that thanks to that effort that we made, today there are 3 vaccines undergoing clinical trials, in West Africa. So we got this published in, we had the front cover of Cell, at the time. So, and then that was very gladly. But then also, applying, metagenomics on site in Africa, we were able to do the first, metagenomic in clinical samples in West Africa. And that really was very helpful because we were dealing with a situation where people presented with some cases of fever of unknown origin. And the idea was to identify the pathogen circulating in those in order to provide a solution. And in the process, we discovered 2 novel rhabdoviruses, and then this novel rhabdoviruses that we call epomavirus 1 and epomavirus 2, were very similar to a virus that was responsible for an outbreak of a viral hemorrhagic fever in the DRC. The virus in the DRC was called the Bakongo virus, but then this is what we discovered in West Africa, and then developing an analyzer assay for this in order to go to the community to see how prevalent this virus was. We found that about 80% of these viruses were, 80% of people in the communities where we discovered that had antibodies. Now, in 2018, for those that actually understand, and follow news on the public health, Nigeria had an unprecedented surge, of Lassa Fever. This is a virus that kills thousands of people in West Africa every year. Within 8 weeks of 2018, we had 200-fold increase in the number of cases. So that is more cases in 8 weeks than in the past 2 years combined. So there were huge questions that were being asked, especially public health questions. Were we dealing with a new variant, or we're dealing with more virulent strain? Was this surge due to human-to-human transmission? Or is it the fact that our diagnostics were either ineffective? So we swung into action, working with the Nigeria Center for Disease Control, but then collecting samples across the country. And then in the first few days, we performed 26 sequences that eventually did about 300, and eventually went on to do about 300 full-length genome of the Lassa virus. This was done in real time in Nigeria, and then we were able to help the public health to respond to this question. We're able to make clear that there was no evidence of particular new virus strain, no evidence or increase of sustained human-to-human transmission, but then that the search was due to multiple introduction of genetically independent viruses similar to non-lineages that we know. That is, identifying lineages 3 circulating only in the northern part of Nigeria, lineage 2B circulating only in the eastern part of Nigeria, and lineage 2A circulating only in the western part. So we had this nicely published in New England Journal of Medicine, but then this really helped us not only to address key public health questions, but also understand Lassa viral population circulating across Nigeria. And what really was important about our finding was the fact that it was mainly and all about rodent. So we understood that clearly, we could actually, that this research was driven by the animal reservoirs, and then we had this nice publication in both in Nature and then in New England Journal. But what this led us to go on and then set up a nationwide, study to study the Lassa virus genetic diversity in rodents. In that process, we found, we demonstrated for the first time, that we identify here, for instance, you look at household samples that were linked with Lassa, in human. So that is rodent sample infecting humans. So we have several cases of this in Nature, and this is the very first demonstration where we show a clear link between animal, rodent samples infecting human, and that really was very clear that this search was done. So we really use this information to inform both the Nigeria Center for Disease Control and then the WHO. So here is kind of shout out by those two organizations about the critical role that we played in this regard. But then we didn't just stop there. We thought about, diagnostics being very expensive. We went on to develop the first, CRISPR, based assay to identify, to detect Lassa fever in clinical samples. And then we went on to develop, a point of care, a point of care test in order to identify this virus. But then what we did in the process is basically to just use technology to develop, guide RNAs and then use, those sequences that were generated to, along with the nucleases to develop a test that can pick up, specifically different lineages of Lassa fever and then turn it down to like a point of care diagnostic test you see over there. So, and then this was highly reported into Nature. So, and then really showing clearly for the first time how we could use this in the African continent and in the context of the country to respond. But this is another case study where we went on, where we had, in 2018, we had children of school age that were dying in the eastern part of Nigeria. Within 2 weeks, we had over 146 children dying. And then all other tests, Lassa fever, Ebola, and other pathogens that we know proved negative. The sample was sent to a lab, and within 48 hours, we were able to generate, full genomes of yellow fever. And you can see the 2018 yellow fever, genome that we generated, all clusters here. So, and it's clear that it was by urban transmission. But then what was different was the fact that this cluster here was different from the previous genomes of our previous Lassa Fever lineages, yellow fever lineages circulating in Nigeria in the past 92 years. You could see that. And this information was provided to the Nigeria Center for Disease Control 48 hours after getting the sample. They declared an outbreak, and within 2 weeks, they succeeded, in containing this outbreak, saving thousands of lives. So fast forward to COVID-19. So when this actually happened, the Nigeria Center for Disease Control and then the Africa Center for Disease Control, knowing the capability of our facilities, got up to us and then put us on a state of high alert in order to receive samples across Africa. And we received the first, suspected sample from Nigeria around around April 3rd. And then within 48 hours, we were able to have a whole genome of the virus sequence. And then we established the fact that yes, this virus, that was the very first genomic sequencing performed on the African continent. But then this virus actually was very close to the virus circulating in Europe or in Switzerland at the time. And this information was actually provided to public health authorities, not only in Nigeria and Africa, to demonstrate that this was imported in Africa and then in the country. But then most importantly, we went on actually to do additional sequencing. And over time, we're able to use, this kind of next train and then use real-time genomic sequence data in order to inform African, not only Nigeria, but other African government, because we're doing this in so many other African countries. And right now, as I'm speaking, we have 55 different lineages circulating in the country. And then that really has been very useful in order to identify hotspots. Now, we use this information again to go on and then develop a SHINE, which is, the streamline infection to navigate epidemics, to develop this assay for COVID. And then within usually 15 minutes, we get, when we have a short viral load, we can have all the data. And then this really has been very useful in the context. We have two versions of this. The lateral flow version. We also have the plate system that can enable you actually to perform these assays. In terms of cost, that's pretty cheap because it's about $3 compared to PCR. We are in Africa, it's about $150. So here, basically, just showing the result live in on live, and then here for the assay that I just told you. But then we've actually also demonstrated that you could do this for multiple viruses and then multiple pathogens, and then develop this kind of assays within a week. So this really gives us confidence that going forward for future pandemic, we'll have the necessary tools and the technology in order to address these problems. But we could also, massively multiplex these assays, in order to develop a multiplex, and then can pick up to about 300 to 400 different pathogens in a single test. And this is what we call the Kylene, the Kylene technology. And then this is really undergoing validation in the FDA. Now, when talking about this, you could see during the first wave of COVID, Africa did very well by navigating that very well. But around November last year, we actually started facing a huge surge. This surge was not only in Nigeria, but across Africa. So the question that was arising was what was driving, this surge. We identified within Nigeria the new lineage, which is a B1525, that was actually, spreading like wildfire. So, and this really was very helpful. So not only identifying this lineage, but we also went on to actually to look at this geographical spread in the country, but then also identify another new variant, which is, the B1318, that also emerged from within Nigeria and spread outside the world. But in addition to these lineages, we found out that the B117 from the UK actually was in Nigeria, but the B1525 that I just described now has taken over that over the years. So these are basically all, recently we identified this, what I call the Indian lineage, that is the B1617, also in the country. And this may be explaining the reasons. Now, let me come down to something that, in order to identify the origins, of all these viruses and then circulating in Nigeria, and especially, SARS-CoV-2. So we really started looking, doing a study on the ecology of bats to look at their viral diversity and the potential threat to sympatric human population in two ecological regions of Nigeria. So, and then we really went on to do this several studies, but we started by what is very simple, and this is done in collaboration with the University of Ibadan and the University of Washington, but we started doing a serological screening of a Nigerian bat, to have some preliminary, I will discuss some preliminary results with you here. So what is very important in this study is the fact that, we've identified, quite a number of bats, that seems to be harboring what I call, antibodies, to SARS-CoV-2. So, and then this is basically showing antibody to the anti, proteins in bats in several places in southern Nigeria. So you can see here in terms of proportion, 28% and then 21%. And then overall, we have about 18.5%, basically that. So from this study, we actually see clearly that, SARS-CoV-2 specific antibodies were constantly detected in the Erilon herbum fruit bats in both southern and then in the center and the west and the central part of Nigeria. And that's really suggests that it's likely cases of, zooanthropognosis in this case. But what we also found is that, 10 samples tested positive for this conserved NNT, antigen, which only suggests that the Nigerian bat may also be harboring other coronaviruses. So that for us, and then an ongoing study that we're really doing now is actually to identify SARS-CoV-2, related corona and other SARS-CoV-2 coronaviruses in real time, using both PCR and NGS and the metagenomics. So really, and they're also looking into, the detection of bat filoviruses. So these are some of the things that we're doing, and then I hope this were able to give you an overview and then short into some of the things that we're doing. So I really want to end this by acknowledging our standing partners, both within Africa and outside Africa, and then our funders, who have been supporting these efforts all over the years. Thank you.
Thank you, Dr. Happi. Excellent presentation. Gave some really nice examples, I think, of some of the challenges in terms of the regional context of disease that's circulating in West Africa. While people are perhaps thinking of some questions to type in the Q&A box, can I just go back to this idea of regional surveillance and regional support? So a lot of the examples I think you provided in the presentation were focusing on Nigeria. How does regional cooperation and regional surveillance activities work in West Africa, and what are the mechanisms to facilitate that?
Yeah, I mean the whole idea of regional surveillance in Africa and in West Africa is actually coordinated by the African CDC, and then they have regional offices across the region. So as I said in the introduction, so we are a WHO and then African CDC, regional reference lab across the region. So there's a surveillance network, and especially what we call the Pathogen Genomic Initiative, by the African CDC. I'm sure you must have heard about that. It's a very huge program. And that really, we have labs at different levels. We have national lab, we have regional labs, and then this is for pathogen genomic surveillance, for instance, and then we also have the reference lab. So among the two reference laboratories, it is my lab and then one other lab in South Africa, which is the KwaZulu-Natal Research Institute, that is CRIPS. And that really, we basically have oversight over all of the regional and the national lab, but we also ensure we develop, we build capacity, and we also create, develop new technologies and innovation, and ensure that we translate some of this genomic knowledge and then and technology, translate them into point of care and then tools that are very applicable, to the African context. As you know, the African public health context is three-dimensional. We do have like the primary healthcare system that has little or no facility, and then as such, tools like the point of care diagnosis that we're talking about here, are actually adapted for those kind of environment. Then we also have the secondary healthcare facility system, whereby some of the diagnosis that I mentioned could be very useful. So we definitely have a bottom-top approach, but work with like the West African Healthcare Organization, the National Public Health Institute, but then coordinated, by the Africa Center for Disease Control, in order to provide, in order to provide the knowledge, the skill, the training, and also leverage the platform that we've established. For instance, my lab has like a NovaSeq 6000. So you can imagine in terms of number of sequences that we could run in a week. So then we pull samples from other African countries that have such facilities, for instance, for COVID, and then generate this data, and then eventually just make this data available to those countries in order to make informed policy and then decision. So we basically sequencing samples from multiple countries across Africa, and then use such information so that I make them actionable, so that they can use information, and then you can use them in order to make public health, decision. So people will be asking questions, why is Africa negotiating? There are so many unanswered questions, but I can tell you that behind the scenes, a lot of these efforts that are going on, and that most African countries are using in order to respond to the pandemic.
Yeah, thank you for that. I suppose following on from that discussion is thinking about animal diseases, which are perhaps even more neglected, particularly across West Africa. And we certainly, for foot and mouth disease, we have some experience of some of the challenges in collecting samples from the field and doing that type of diagnostics for animal diseases. So perhaps a maybe ask you $1 million question, or perhaps it needs to be $1 billion question. What do you see as the key activities that are required to strengthen animal disease surveillance across West Africa?
Yeah, I do think that, zoonotic surveillance is going to be and then as for the animal disease surveillance is very, very key. And then as such, there is a need, to have, to transfer some of these technologies that I heard the people and NBAF talk about, and then FARO in the US, and then the USDA, the USDA. So I do think that, we recently signed an agreement with the USDA, and then which is FARO. So, and this really will be very important in terms of, not only transferring technologies, but actually leveraging the facilities and then the skills on the ground in order to set up a surveillance system, that will really serve as part of what we call the early warning system they want to establish in order to bring together both animal and then human, zoonotic diseases together, because this is going to be a key activity in order to support, not only animal disease, but also to support the global system.
Thank you. There's a couple of technical questions from the floor regarding SARS-CoV-2 in bats and the data you presented in your presentation relating to that. So one question is, does SARS-CoV-2 positive BAC sera neutralize SARS-CoV-2? So does it cross-react with the human sera, the human viruses, sorry?
Well, I do believe that the assay that we use, that was one of my postdoc in my lab, that is George. From the result that we have, I think they cross-react.
And then a follow-on question, which is thanking you for the presentation, but what extra precautions were taken when testing bats for SARS-CoV-2 to avoid asymptomatic transmission to technicians and scientists working in the lab? And I suppose this is a sort of a generic question. I think that all of us working in labs working with SARS-CoV-2 over the last year or so have had to deal with.
Yeah, that's a good question. Alice, we've been working in this space for over decades, and then definitely we'll take all the necessary precautions, as you mentioned, even in the field. If you see one of the slides I was telling you about rodents, so we're fully kitted, all the full complement of PPE, and then making sure that, the samples are actually taken up from the field, but then they are brought to the lab for additional analysis. So we take full precautions. So, we're equipped with the BSL-3 facility. We make sure that we do things within the within the biosafety, the necessary biosafety containment, and then making sure that we also respect all the necessary safety measures.
And another question, perhaps the last question from the floor relating to your presentation is you discussed the CRISPR-based detection methods, the Sherlock assays. I had a question myself, which was something about what are the advantages of Sherlock over other field-based nucleic acid methods such as LAMP and RPA that are widely used. But I suppose a similar question from the field, which is do you envision the new CRISPR-based detection methods as having the greatest impact in point-of-care uses or through lab-based detection, especially for novel infectious agents? i.e., will it be cheap enough to deploy more broadly?
Yeah, we believe that it's going to be cheap enough. The recent version of the Shine assay that we actually developed is actually better. It's actually a room temperature. You need no equipment, you need no facility. Everything is at room temperature, and it works so well because of the temperatures in Africa. So, and then in terms of cost, I think right now we're probably about $4 to $5, and then I'm sure with time we should be able to drive down the cost further. And that's the reason why we're really spending so much energy and then spending time on this technology, because we believe that is well suited, for Africa, for instance. So really do not need, it's not going to be, the point of care here is just a lateral flow assay that does not need, to be in a laboratory. It's something that could be done in the field setup. And then in places, where you really do not need a lot of high quality technologies and facilities. Now, in terms of developing this assay for different type of pathogens, so we demonstrated that we can do that for many pathogens. I gave you 2 examples, Lassa. We also have that for dengue. So we've shown clearly that, and in addition, we have the possibility now to multiplex this, and then do multiple CRISPR tests, and then that's really going to drive again further down the cost, and but also creating a situation whereby you could have a one-stop shop where you could diagnose multiple pathogens in one single assay and see how you can quickly respond to potential emerging emerging diseases.
Thank you. I think we'll have to close the questions now, but thank you very much for answering the questions. And like I said earlier, we'll pick up on any remaining questions in the round table discussions later on. So our final talk today is from Dr. Ethan Jackson, who is a Senior Director and Principal Researcher at Microsoft Health Futures. And so Dr. Jackson directs Microsoft Premonition, or Premonition, yeah, an advanced incubation product aiming to detect the movements of potential pathogens in the environment before they cause outbreaks in humans. So his talk will tell us a little bit more about Microsoft Premonition, which is a sensor network and data platform for biothreats. Thank you. The floor is yours.
Wonderful, thank you. So really briefly, I am part of a group at Microsoft called Microsoft Health Futures. You can think of us as sort of the moonshot, the health moonshot parts of Microsoft. We can come back to that later. All right. Okay, there we go. All right, so I guess we're all here for this, the same reason, which is that the Earth's biome, that is the set of life all around us, impacts the health of every society, economy, and government. And what we all really want is exponentially more data about the distribution and evolution of its species. Because if we don't have more data, it's going to be difficult to predict and prevent disease outbreaks and epidemics. It's going to be difficult to predict and prevent invasive species and agricultural pests. And it will be difficult to manage our biodiversity and ecosystems. The challenge as an engineer is that most species are invisible to the billions of sensors deployed across our planet. I mean, it's pretty incredible from the engineering point of view to really think that as a species, we have successfully deployed billions of sensors that are able to detect all sorts of things. But from the perspective of the biome, most life is invisible to these sensors. And the bar chart below shows kind of the reason why. Most animal species are arthropods, about 10 to the 6 species, millimeters in size. And most species are bacteria and viruses, microns and nanometers in size, estimated to hundreds of millions to billion species. Nobody quite knows for sure. So this is basically the engineering challenge. How do you build a sensor network that is able to see the movement of these things in the environment? And that is the goal of Microsoft Premonition, is to really sort of reflect on what technologies are available, what technologies need to be built and combined so that we can continuously monitor the biome. Our focus is on providing real-time data about viruses, microbes, and arthropods, because that comprises most biodiversity. And so how do we do this? From the engineering point of view, the first capability that we think is really important, because it's not realistic to test every nook and cranny of the world all the time, is low-cost digital sensing that can detect some key biological threats, but also allow us to sort of autonomously prioritize sample collection in molecular diagnostics. And so for this reason, we've started with the notion of robotic smart traps that focus on terrestrial arthropods, which comprise most animal biodiversity. The goal of these smart traps is to autonomously identify, continuously observe, and then selectively capture terrestrial arthropods. From the human health and animal health point of view, of course, these are disease vectors. From the agricultural point of view, they're pollinators, pests. From the biodiversity and sustainability point of view, they're bioindicators. And of course, from the kind of sentinel detector point of view, they include things like blood-feeding insects. So a bunch of important species, if you really think from the perspective of what would a sensor network need to do to help prioritize our monitoring of the biome. Let me just share with you, just to put meat on the bones here, what our first generation robotic smart traps look like. You may have seen these before. This is a cylinder of an array of these kind of smart cells. Here's a zoom in of one of these smart cells. And the way they worked was by emitting an infrared beam across this opening here. And when an insect flies past this infrared beam, their wings occlude this signal at a high frequency. So basically we're measuring an optical wing beat. And it's been known that wing beats can be used to speciate. And so when something flies past the sensor, we get a biometric signal on it in real time. On board this device, in real time, we're attempting to perform a classification. And we will record what that wing beat looked like, what we thought it was, along with additional abiotic data, the subsecond of encounter, ambient light, barometric pressure, all of these additional covariates that might help us understand what's going on in the environment. And then the second thing this device can do is it can make a decision. So when something crosses this beam, it can decide that it would like to capture it, in which case this door would snap shut and a specimen would be trapped in the back. So in terms of the data this provides, it's providing real-time data about all of the insect encounters, presumably flying insects in this early device, and then it's subsampling a set of biological specimens. We first deployed this in Harris County. We were building the system, and it just turned out that it was the time that Zika may emerge in the US. And so Harris County, the county which is home to the city of Houston, asked if we would be interested in collaborating, allowing them to provide, working to provide more information about the Aedes vectors of Zika. So here you're seeing a night vision camera looking at this early prototype device. You can see the infrared LEDs here that are creating that light plane, and that's Aedes aegypti kind of buzzing around. If you watch in the center here, you'll see a door snap shut and collect one of these aegypti. I'll just let that repeat. Okay, there it goes. And so the interesting thing to call out here is that decision was made in real time by machine learning happening on the edge here. So it's not just a simple beam crossing, but it's that this thing was programmed to look for things that were Aegypti-like based on their wing beat signatures and then capture that sample. From the vector biology point of view, on the left there, you see an actual field collected Aegypti in early experiments that we did in Harris County. You can see as a consequence of this design, you tend to get high quality morphology, which really enables supervised learning loops from the machine learning sense. What I mean by that is that if you take this out into the field, you bring it back, you have a one-to-one mapping between a morphologically intact specimen and the digital data that was collected. And that is just kind of perfect for training up a machine learning system to start to differentiate. In fact, we found that with a few hundred samples, we got pretty accurate, even with this early design. Accurate meaning about 90% accuracy to distinguish wild-type Aedes aegypti from wild-type Culex quinquefasciatus in Harris County. Now, just fast forwarding a few years ahead, I'm not going to talk about our new device designs here, but I'll just give you a sense of what our V3 optical wing beat sensors look like now. So this is what these sensors will detect in real time to make those putative IDs and to make those sort of autonomous decisions. These are spectrograms. Here you see a wild-type Culex restruens, wild-type Aedes albopictus, and a wild-type Sphaerophora columbiae. So today we build up this library by bringing wild-type species to Microsoft, and then we start to build up, the spectrogram so we can train our machine learning systems ahead of time before we deploy systems back into the environment. Of course, in addition to identification, what we also realized is when you aggregate these systems together, because the data they collect is all real time, you start to be able, it enables you to create these high fidelity models, presumably for public health. So in this example, you're seeing models that we were able to create back in 2016. The greenish lines represent Aedes, the orangish lines represent Culex, and the x-axis is time of day down to the minute. Each graph is a model of what we would expect Aedes and Culex to do based on this continuous streaming data where IDs are autonomous and information is sub-second. So this is a learned model of overall circadian rhythm, for example, how these species react to light levels, temperature, humidity, and so on. This is trained using kind of modern Bayesian mixture model techniques. If you add all of these predictions together, then you start to recover the complex, nuanced behavior that you would see, that an individual trap might see in the environment. And obviously, what becomes interesting from the public health point of view are the places where there are peaks, the places where you may optimize an intervention. If you saw this for a while, you may be interested in where there's an anomaly, the presence of something that you would not normally expect to see once you have these kind of high fidelity models. So in summary, on the sort of zooming back out to this notion of a network, a sensor network, a network of robotic smart traps enables some important capabilities. Of course, it enables species identification at each node, which is a key bottleneck in the surveillance of vector-borne diseases, because you need to understand the composition of vector species. But when you have a bunch of these, now you start to be able to aggregate these and provide new sorts of forecasts, because you can see this temporal high resolution data across many places. And then from here, you can imagine doing a few things. One is to use these real-time forecasts to apply interventions earlier. But the other is to use this information to now filter the environment, to look for those things that you do want to spend the extra effort on to zoom into them, possibly to look for new pathogens, to look for explanations of insecticide resistance, to detect invasive species. So the other, I think, important thing is view this as low-cost digital sensing, which is allowing a filtering of the environment to try to provide specimens for information maximizing diagnostics. And this is all sort of mission programmable now. The whole thing is dynamic. And so that brings us to the second capability that we have been working on, and that's a capability to estimate the biological composition of what you have prioritized to sample from the environment. And of course, because so much of the biome is known, is unknown, we need to be able to detect both known and novel life forms. As described before, our approach, because it allows us to see across all of the taxa of life, is to use metagenomic analysis. Again, higher cost sequencing compared to the low cost digital sensing I described is really important, because it allows one to prioritize what samples go through this process. But once you have done the sequencing, we want to be able to recover known and novel viruses, known and novel vectors, environmental microbiomes, vector host pathogen associations, drug resistant genes. On the left-hand side, there is a Petri dish with a mosquito in it. We would like to be able to reveal all that we can know about that mosquito once it goes through a sequencer. In this particular example, in that Petri dish is Anopheles stevens eye. The blood meal in there is a gorilla, not a human. This was, of course, a lab-created mosquito. But the point here is that we ought to be able to figure out that overall set of associations that were physically co-located in that sample. So first, as was described earlier, the goal here is to turn DNA into data. From our perspective, this is more and more field deployable. The pipeline I'll describe both works with Illumina, but it also works with field deployable sequencing like Nanopore. So just for those who are not that familiar with the sequencing process, I think the cool takeaway here is that technology is progressing to a place where sequencing no longer requires a large facility, right? That we can really do this near a network in real time. The second thing we need to do is to take that sequence data and then analyze it. And this is where our pipeline is a bit different, maybe from the ones that were described earlier. What we do is we take in the output of a sequencer, and we will map it against all partially sequenced species. So right now, that's a database of about 3 trillion nucleotides, and that covers all taxa. We'll do this by matching each little bit of genetic material against this entire database, allowing a pretty large divergence from the genetic snippet and the reference. And as a result of that, we will get this gigantic set of possibilities. Most of them will be wrong. That will require cloud scale analysis already, because to do that with inefficient algorithms, would take 4 or 5 years, depending on some estimates. So you need cloud scale. And then the second thing you need is a machine learning approach that takes all of those possibilities, looks across them, and then builds a statistically most likely model. That's what reduces false positives and then bins the genetic material in ways that is useful. From this binning now, you can start to see signals that you didn't know you should be looking for, because we're taking into account all taxa. And for more specific questions, like what bacteria are there, what viruses are there, now you can target more expensive algorithms, because you've taken this complex environmental sample and you've binned it. And then you want to do things like look at this data set and figure out the needles in the haystack. So just a quick view here. This is a large data set we analyzed. This was the Anopheles Gambier 1000 genome data set. created for the purpose of Anopheles gambiae population genomics, but we did a metagenomic analysis of it. So it's a pretty large data set, 100 billion reads. In each column here, you have a mosquito from this data set. In each row, you have a species name. And each cell is glowing based on what's the probability that we estimate that species to be in that mosquito. This, as I mentioned, is a sound probabilistic model. So it does not make a hard call, it makes a soft call. And so, for instance, you can see in this data set, where you have species that are very similar, Anopheles gambiae and Anopheles colucii, it is giving you the option that it's possibly one of these two. So you really should understand that as a probability. And then as you go down the list, you will see things that are related to host, environmental bacteria. So all these signals start to kind of pop out when you can do this broad spectrum analysis. Let me give you a really quick example of kind of finding a needle in a haystack now. I think I'm doing okay on time. Is that right, Don?
You've got about another minute or so. Okay,
great. So in this case, what we're going to do here is take this 100 billion read data set, try to quickly find a needle in a haystack. What kind of jumps out in this data set is the presence of cow. So we're just going to create a little filter here where we'll filter this data against the genus that contains cows and include all viruses. And then just kind of replot this diagram. So I should have accelerated this little demo for timing purposes. Okay, here we go. So filter 100 billion reads against this probabilistic model, zoom in on what might be associated with cows. We get a new heat map here. In this case, this is probabilistic number of reads mapping to species. So cows are at the top. We'll just resort this thing. So we go in descending order of cow signal. And then everything else in this map is presumably a virus of some sort. I'm going to pick one that's glowing a bit brightly, right over here in the top left. I'll see the reference that we mapped against. I'll click on that reference. That will take me to a link to NCBI. And then here I'll see that this was a bovine parvovirus. And so that's how we discovered a bovine parvovirus in a cow. Presumably the mosquito was an incidental interaction in this data set. So brief summary there. At this point, we've analyzed about 80 trillion base pairs of complex environmental samples. The more complex, the better. Some of our collaborators are on this call, so thank you for that. And we're really interested in making sure this is a broad spectrum capability that spans sample types. So mosquitoes, ticks, classic reservoirs of zoonoses. As I mentioned, core reference database is 3 trillion base pairs. This is where you really need cloud scale. That's an expansion of about 230%, including as we learn more about bacteria. And in terms of needle in a haystack, we want to be able to efficiently find those needles. This bovine parvovirus is a 5.5 kilobase virus fished out of 100 billion read data set, and we didn't even know we should be looking for it. So big picture, and I'll maybe stop here. The big picture is, I think, there will be new networks that can map the biome and detect biological threats in near real time. The approach that we're taking is to start with a network of robotic smart traps that can really cooperatively learn to lure, identify, monitor, and capture arthropods. We think that's important because that low-cost digital data is relevant to a bunch of disease systems without requiring the high-cost sequencing upfront. But as you start to see what is in the environment through low-cost digital sensing, you now have a way to filter and prioritize. For us, it's very important to think about how do we preserve chain of custody of these samples? How do we preserve the whole environmental nucleic acids that may be associated with them? But once you have that prioritization scheme, then we really want to do a full broad spectrum metagenomic analysis that's looking across all taxa. So we get as much information as possible from that sequencing activity. So I will stop there for now. The rest of my slides are kind of how we plan on launching Premonition, but maybe I'll just pause here for questions.
Thank you, Dr. Jackson. Very fascinating presentation and some really nice, interesting ideas, I think, about how we might start looking for novel pathogens, perhaps in a slightly different way. So while others are typing questions, perhaps I can ask a question as a non-entomologist, and so I can ask any questions, I guess, here.
Yeah.
And so,
I'm not an entomologist either, so we'll see how it goes.
So if I'm understanding correctly, the device will capture a wide range of different vector species. Are there any limitations at all on the range of vectors that might be captured? I mean, is it completely universal, or are there some gaps?
Yeah, it's a great question. I would say we don't know. And for existing traps, what exactly is the bias that trap has? It tends to be a difficult thing to figure out. The approach that we've taken is twofold. One is to make sure that our systems are programmable. So that means that their lures, the way they breathe out CO2, the way that they produce heat, the way that they produce different spectra of light, is something that is fully adaptable. It's not in the design that I showed you here, but it's in our next generation devices. And so that adaptability allows essentially these robots to sort of fish the environment in a dynamic way. Now, what is the bias when you take into account that adaptability? Again, we don't know. We've started to study that by going to places where we'd expect to see a lot of biodiversity. We did biodiversity studies in the Florida Keys, where you have these interesting island chains. And then we also did a study in Tanzania, kind of right on the border of Lake Tanganyika, where you both have a diversity of vector species, but then a bunch of other arthropod species as well. So I guess short answer is, we believe that programmability and adaptability of the platform is key. And it will take a lot of information to really figure out what is the overall bias, what does it get you and what does it miss? Yeah, good question.
So in terms of biases, a sort of another slightly, again, as a non-entomologist, a technical question is, how many insects, how many individuals is it capturing? And based on the likely occurrence of those pathogens in the insect populations, can you draw any conclusions about the likely depth of surveillance that you get using these types of tools?
Right. So in the devices that I showed here, we had just 64 capture cells, so we could maintain this one-to-one mapping. And our next generation systems, we'll be able to essentially collect and aggregate a bunch more samples. So I guess in terms of your question, how do you decide to collect? I think that as we deploy these systems into the field, this now becomes much more of a machine learning question. The idea is that you don't have to set that definition up front, because what you will see as you set up sites is the overall sort of population structure of vectors. You'll be able to superimpose that with information about sort of host ranges, other environmental covariates. And then what you can do is essentially define an optimization function that lets this network probe. From the machine learning point of view, there's kind of two ways we think about that optimization function. One is exploiting the information you already have. And so that would mean if I know that there tends to be hotspots over here, and I tend to know that the vectors are that, then I will make sure to get a reasonable amount of those. But the other one is exploration, that I will go off of what I know, because I may discover more information by doing something that is different from what's to be expected. And so in the context of a sort of network-wide machine learning loop, what you want to do is provide the sensors and the lures and then the aggregators so that feedback loop can be now dynamic. And it's a very different model from kind of what is out there today. Yeah. So, but we'll learn as we go. The system will learn as it goes, actually.
Yeah, thank you. And so there are a couple of questions from the floor, if we've got time. So a lot of people are saying this is awesome and they really like the technology. One question is how rapidly are the data that you are collecting becoming public, so that entomologists and other scientists can focus their research on the details of your on the details of the work, and perhaps for those scientists who don't necessarily have the time, energy, expertise to examine everything?
Yeah, it's a really great question. So I guess there are two answers to that. It depends on where we are. When we collaborate with academic labs or public health, we will, we will enter into specific data sharing agreements that are appropriate for those collaborations. And so that I think kind of has to be the way that it's done. I will say what we announced in 2020 through our partners with Harris County is that we will establish a Harris County Healthy Biome Network this year. And that will represent the installation of these sensor networks throughout Houston with the goal of longitudinal data collection, allowing us to really understand how these systems can do things like AI-enabled suppression of vector species. And so that will produce a large data set. And one of the things that we are discussing with Harris County now is what is the right way to share that data set. This is actually also in conjunction with the National Science Foundation. They run a program called the Convergence Accelerator, which is asking how do you bring public-private partnerships together to accelerate solving societal challenges. And so we also are very fortunate to have the NSF as a partner here to help us think through what is the right way to do that, that both respects privacy, respects the public health process, but also enables science to be empowered. So I think there's no simple answer to it, but it is something that we're trying to think about very carefully.
And sort of to follow that on, in terms of scientists that are interested in accessing the technology, is the technology commercially available? And so there were a couple of questions on that topic from the floor relating to certainly to the hardware, but also to the database. Are there opportunities to access and work with you to get the database, or do you imagine that those types of relationships are set up on a one-to-one basis on a sort of a collaborative under collaborative frameworks?
Right, it's a great question. I would say we're doing a combination from the, if the question is from sort of the academic point of view, I would say just reach out and let's figure some stuff out. What we did announce in September of last year was our early access program. And that's a program that Microsoft does for emerging technologies. The goal of it is not yet the sort of cost optimized, broadly commercial off-the-shelf things, but rather if there are some scenarios like how do we digitally transform a city? It is through those programs that we can now really scope out a larger kind of foundational question and then make sure that we are able to deliver the whole portfolio to achieve that. So it's kind of a vague answer, but not off the shelf yet, but we are able to support a range of collaborations. The ones that are most strategic for us are the ones that are really getting us closer to what does it look like to have a large network that allows you to in real time monitor and then triage and prioritize what you sample. So if folks on the call, if some of those are jumping out to you, would love to follow up. Yeah.
So I'm guessing based on the responses here and the questions and the interest, I think you can expect to receive quite a lot of emails over the next few days. So the last question really, which is one I totally understand, is so when you describe the pathogen discovery based on the cow virus, presumably the mosquito had been fed with cow blood.
No.
No.
I see. Right. Yes, the mosquito was presumably bit a cow. Sorry, when you said fed with cow blood, I thought you meant a lab experiment.
No.
Yes, mosquito bit a cow. So in that case, you're seeing the arthropod itself as a sentinel, which is something that we have always been interested in. Beyond the direct, phenomena where arthropods vector diseases, they also interact with the biome in a whole bunch of complex ways. Does that allow them to be a sentinel into the larger biome?
Yeah. Okay. Well, thank you very much, and thanks for answering the questions. So, if I can bring this session to a close. So we've had the 4 excellent presentations, and so if we take a break now for approximately 15 minutes. We will reconvene the panel and have the opportunity to ask questions and to continue the discussions relating to the subjects and the talks that we've heard previously. So everybody on a break, please join us again in a little bit less than 15 minutes. Thank you.
Thank you.